Hi, I'm Toby Holda with the Illinois River Biological Station in Havana, and I'm going to be talking about current status of fisheries and aquatic vegetation communities at the Emiquon Preserve. First, I want to say thank you to all of the people that have made this possible. I want to thank the Nature Conservancy for funding and supporting and allowing us to go out and do the work on the Emiquon Preserve. I thank the Illinois River Biological Station for giving me the opportunity to do that, along with the history survey. I particularly want to thank all of the technicians that have put a lot of time and effort. They put most of the hard work into getting the fish, getting the nets, working with the vegetation out there on at Emiquon, and have to put up with my personality and all of that type of stuff. I do want to especially also thank a few staff members that have come out to help with that from IRBS, and a big thank you to the TNC staff that have been super supportive and have helped out a lot. So I want to start by talking about water quality at Emiquon. It is an aquatic habitat. That's what we're focused on, and the water quality matters a lot. Oxygen content has been pretty stable and pretty high throughout most of the time of our monitoring at the preserve. Last year's data shows a big spike, and I'm not sure if that's an artifact in the data. Definitely the percent saturation is going up, but yeah, I'm not sure why that is, but generally speaking, oxygen content has not been a concern at Emiquon. What has been a concern is decreasing water clarity. This is a little bit of a difficult plot to absorb at a glance. I've looked at it quite a lot. So what you have is each line from the top to the bottom is a year. So going, I guess the line across horizontally is a year. And in that year, showing the distribution of water clarity values divided by the depth at which that water clarity was taken. Ideally, you want the Secchi depth to be at least the depth of the water column so that light can get down there for plants to grow. Sometimes it's taken at a nearby location to where the sampling's happening, and that way, we can have depths of a little bit more than one. The goal is for that to be at least half, because then it can get down all the way to the bottom for plants to grow. And you can see that over time, which is as you move down this graph, the distribution shifts from the right to the left. We get more red values, which is light is not getting all the way to the bottom, till at the bottom of the graph, we only have 20% of our instances of taking a Secchi depth reading where the light would actually be reaching the bottom. Okay. All right, so I want to shift to talking about the vegetation community. We spend a lot of time in August and September each year monitoring the vegetation community. In previous years, going far back as 2015 and further back to the origination of the monitoring program in 2008, vegetation monitoring crews were out at Emiquon from April, or maybe it was March, but through October, and starting somewhat early in the year. Recently, since 2016, we've only been going out in the peak growing season to capture that peak biomass of vegetation, that peak coverage, and shifted from using the rake sampling method, which is employed by the LTRM crews throughout the upper Mississippi River Basin in 2015, to using the box method in 2016. I've gone over this before, so I won't spend too much time on it, but briefly, the rake method allows us to take rapid sampling across a lot of spatial coverage in a single day, whereas the box method takes a little bit more time to get to a sample. You get a smaller area sampled at that site, and then that data comes back to the lab and takes a little bit longer to process. I don't actually have box results yet because I haven't gone through the frozen samples from last year, but I do have rake results. One other difference is that with the rake results, we get rake scores, whereas the box results, we get actual dry biomass estimates, and that's able to be converted to a unit of biomass per area because we use an aerial sampling tool. So looking at the long-term abundance of vegetation at Emiquon, we can see that it got high very quickly, which was one of the great restoration success stories. Not any plantings were needed. Vegetation came back with I was going to say vengeance, but that sounds negative. It's a really positive thing. And you can see from here, the amount of that was native was really high. So that was very encouraging. Vegetation levels were high for a long time. Again, there's this gap in the rake data. But you can see that it used to be very high, and in the past couple of years when we've gone out there to do more rake vegetation, abundance is low. Now, we also know from data from Forbes biological station that have been doing cover mapping, and just from being out there in the system, that the amount of area covered by vegetation has receded a lot. This is not what this data is showing. These data are showing how much concentration of biomass there is at a site where vegetation is present. So what we're seeing here is that not only what Forbes sees that the receding of the area of biomass, but here that density of biomass is less when we have it, which can be because it's growing in shallower water, so it can't get as tall because it has to grow in shallower water where light can reach the bottom in the turbid water. Or it could be because it's just more sparse when it is out in deeper water. Both of those things seem to be true. Looking more carefully at the community changes over time, throughout most of the history in the first several years of the program, it was dominated by, I'm going to go from the bottom up here by color. We have Eurasian milfoils, the green, southern naiad is the orange, the purple is American or longleaf pondweed, the pink is coontail, and those are the players that we still have a lot of. The green is sago, we still see some of that. The brown is Elodia, and then there's some brittle naiad in there. So that's mostly what we used to see. There was lots of all of it. Now we mostly just see the milfoil and either the southern naiad or the longleaf pondweed with a bit of coontail mixed in. I'm going to zoom in on the last 2 years because they're so shrunken here. We'll get a better idea of what's been going on in the past couple of years. So from 2023 to 2024, there's a change here with an increase in Eurasian milfoil species and a decrease in southern naiad, but also an increase in longleaf pondweed. We also saw some native milfoil in 2023, which we knew at the time could be hybrid, and based on the fact that we don't see them anymore, they either got eliminated or were actually hybrid, and now they're just showing more of that Eurasian milfoil character. One of the things I note here, when we were out at the preserve, it did seem like vegetation had expanded, at least in terms of area, so I'll be eager to find out from Forbes if that's what's been happening. But here you see a reduction in rake score, and it did seem like a lot of the areas where we had vegetation that we didn't before, what we were seeing was an expansion of sparsely distributed milfoil, which would explain why we would see more milfoil overall, but why the scores would overall perhaps be lower. Looking at the box data, again, I don't have data for 2024 yet on box vegetation abundance, but we do see, starting back at that same color schemes here, we see the basic presence of Eurasian milfoil and coontail and sago, and we see that somewhat diminishing over time. We see a lot more representation of longleaf pondweed, but that also dropped out over time. And so in 2023, it was more similar to what we were seeing in the rake data from before 2016 and starting in 2023 as well, with the dominance of milfoil and southern naiad, but also with some sago. We included algae data in that year because it had been included in previous years, but generally that's not part of the submerged aquatic vegetation survey. So, okay, so now I want to talk about the fish community. That's what we spend a lot of time out there doing. My background is more fisheries oriented, and that's definitely something that can have a big impact on the community and on the ecosystem, and is very important to users of the preserve as well. We sample using LTRM methods from April through October. Long-term, I think I mentioned that before, that's the long-term resource monitoring program run through the Army Corps and the USGS. That's done again throughout the Upper Mississippi River program. So we'll use fyke nets, which is what's used in backwaters. These are large fyke nets, which catch larger fish like gar and crappie. Smaller fyke nets, which catch a lot of young ear fish, some of the larger fish. And then electrofishing, which catches pretty similar species. So over time, abundance has been high and seems relatively stable in terms of numbers. Over most of the time period of the study, abundance has been primarily native species. There is some increase in non-natives, but that's primarily in terms of biomass. And some of that currently in our monitoring data, that's all common carp. So it's an increase in the biomass. So the carp are getting bigger, not really that much more abundant, but a lot bigger, at least relative to the rest of the fish out there. There are also silver carp and some bighead carp in the system, but we don't really capture those in our monitoring data. Okay, so there's a lot going on here in this graph of community data. There's a lot of abbreviations and different colors going on. I'm going to just highlight some basic trends over the whole time period and then in the recent couple years. Overall, this system has had a lot of bluegill and gizzard shad and largemouth bass. In more recent years, it's had a lot of yellow bass by abundance. And that's just kind of what we see. I'm not going to spend a lot of time on this figure because I think biomass is a more important metric to be thinking about. I will say, there's this huge number of gizzard shad. We'll see that drop out in biomass because a lot of those were part of a particular minifyke. We had over 1,000 really small gizzard shad show up in one of our nets. Okay, so again, similar importance of species. We have bluegill and gizzard shad. The largemouth bass are more important in biomass, of course, because they're bigger. We do see yellow bass, and we see bowfin a lot more prominently, as well as common carp, which is that second green section closer to the top, showing up as well, and some green sunfish. In the last couple of years, we've seen declines in largemouth bass abundance, some increase in white crappie abundance, which is that purple section, and in carp abundance, which is that upper green section, and some decrease in bowfin biomass. This shows the largemouth bass catch per unit effort over time, which is pretty similar between random and just all sites together. This is from electrofishing data. You can see, it used to be somewhat high. It's gotten less in recent years. Just to give this some context, we're going to compare it to data from around the region. So right now, the only thing that's for Emiquon is the red line, which kind of starts high, bounces around, very similar to Pool 13. In fact, I was looking at this and noticing just how synchronous those two are, which is kind of striking and fascinating when it's really next to Lagrange. So there might be some regional level factor that's going on there. I'm not really sure what that would be, but that just stands out. But the main thing to notice is that the bass at Emiquon are similar to the abundance of bass, largemouth bass in Pool 13, which is considered a nice quality largemouth bass fishery and are much better than the nearby Lagrange Pool backwaters, or even the Alton, the section of the Mississippi River just above Alton largemouth bass community until the past 3 years. The past 3 years, abundance of largemouth bass at Emiquon has been very similar to those at Lagrange backwaters. So we're kind of losing our advantage when it comes to this particular sport fish. Gar and bowfin have been pretty healthy and increasing over time. In the past 5 years, gar have kind of come down from this peak abundance, and bowfin seem like they might be doing something similar, or maybe they just peaked really high and then have a new normal that's more abundant. Although they have been showing lower weights recently, so that may be a cause for concern. Okay, I want to talk about the relative weights of the centrarchid species. We look at this every year. We haven't really expanded to other species much yet. For the most part, the fish at Emiquon are doing pretty well. Relative weight, pretty consistent. The black points here are last year's data, and all the rest of the points are all the previous year's data. Pumpkin seeds are smaller, not for their size. We just have smaller pumpkin seeds than what we used to have. The x-axis here is the expected weight for the length, and then the y-axis is the observed weight for the length. White crappie, again, they're similar to what we would expect. They're not that, but they're a little bit higher than what we might expect. A little bit higher relative weight compared to their standard weight. And the sizes of white crappie that are being seen are larger than what we've mostly seen in the past. So they're growing larger currently. And just kind of to illustrate that again, here's their length versus weight. In this one, all the black points are what's happened historically. And 2024 is a lot of collection of white crappie, and we can see much larger white crappie than what was before. It's not really different from the equation, a little bit higher, but it is larger animals. Here's a trend over time. Again, the fish are really doing pretty well, hovering around 100 pretty well. You can see that crappie are increasing a bit over time. I didn't show pumpkin seed because we only had a few from last year. We'll come back to that about white crappie later. So yellow bass have been increasing. This has been definitely a major thing. We noticed in the biomass figure that their biomass has gone down, but this is definitely, their abundance is still going up, and so that indicates we're getting a lot more smaller yellow bass. I'm not sure if that's a good sign or not, but they definitely have had an increasing and major impact on the system in just about 10 years or less. Common carp are the big concern right now, and they've definitely been increasing. We definitely saw an increase last year. This is biomass, so we're hoping to be able to track them and probably remove as much as we can. But we definitely see them increasing, so they're not just going away as a threat on their own. And they may be part of the turbidity story, where turbidity has been increasing and water quality has been declining. So I want to just summarize the fish community results. This kind of integrates some of the other things. We've seen an increasing white crappie and common carp. Common carp are related to turbidity in a lot of systems and the loss of vegetation, which is what we've been observing. Interestingly, white crappie are actually more likely to occur in a system compared to black crappie that's got more turbid. Black crappie are slightly more adapted to structure and clear water environments, whereas white crappie are slightly more adapted to turbid and just kind of open water environments. We see decreasing largemouth bass, maybe gar and bowfin. That's not something we like to see with our piscivore community, and also with largemouth bass being a sport fish that people like to catch. We are seeing smaller and more of like the gizzard shad that might have just been a spawn that we happen to catch, but definitely with the yellow bass and black crappie. So again, this all comes back to probably seeing a more turbid environment. All right, and if there's time, I know I've gone a little bit long, I'll be ready to take any questions.